Speak Student!
Molecular genetics, transcription versus replication, a la Shmoop. Okay, we're back with Shmoop Biology, and we're talking about transcription and RNA processing with Dr. Ruth. So what are some similarities and differences between transcription and replication? Like, what are the key things we have to remember as they relate?
Sure. So similarities, they both start with a DNA template. They both involve a polymerase, so either DNA polymerase or RNA polymerase.
Mm-hmm.
They both form new molecules that are composed of nucleotides. So those are some of the similarities. Differences, I think the main ones, one is that DNA replication happens over the whole genome, right? Like all the DNA needs to be replicated when a cell divides, whereas only certain genes get turned on and transcribed at certain times. So for transcription, you need the polymerase really needs to know where to start and where to stop, whereas for replication, it's kind of just do the whole thing.
So that's really interesting. So this is in the 23 chromosomes, it's going to hop, and there's some that are just not lit up when it's transcribing?
Yeah. There are tons of genes on each of our chromosomes, so there's probably a few on each chromosome at any given time that will be turned on. But, if a cell is dividing, a certain set of genes will be turned on. If a cell is kind of just sitting there, then it's going to be a different set of genes.
And it just skips over it, like you're in the express, not the local train.
Exactly. Yep.
Okay, cool. All right, another $5 word. What is a poly-A tail?
Yes. So that's another important difference between transcription and DNA replication, which is RNA gets processed after it's made. So you go through the process of transcription and you end up with an RNA molecule that isn't really mature. So you need to get some changes to it. And one of those changes is you have to add on a tail to the end. It's a bunch of A nucleotides.
So these are just placeholders so that everything matches up sequentially?
It's basically added to the end. So it doesn't really need to match up, but there's just a sequence at the end. To be honest with you, I have no idea what those do. Sorry.
Okay, then it can't be that important. PhD here, don't worry about it. Okay, and that's it for Lesson 7. We're going to move on to the next one. What are some similarities and differences between transcription and replication? What is a poly-A tail?